Hi, I'm Peter Cook. Welcome to the pan-genomic model, the role of transcription factories in chromosome structure and function. I'll begin with an overview. I've divided this presentation into 7 sections, and I'll end with an 8th where I'll answer some frequently asked questions. So the fields I'll be covering are huge and fractured. We've all become so specialized and we work in our own silos that it's very difficult to understand what is going on in other fields. Nevertheless, I'm going to try and seek universal principles applying to all forms of life. My focus will be very much at the level of transcription, and therefore I will be saying little about replication, repair, recombination. And I will try and offer a comprehensive and unifying view, which I believe to be the only one that is currently available. So let's turn to the key principles. There are just 3 formal reasons why a gene might be off in one cell type and on and transcribed in another. The first is that the covalent structures of the two might differ. Here, the lollipops indicate methyl C groups around the promoter, which when present ensure that the gene is off. Otherwise, it's on. Similarly, in the immunoglobulin genes in certain cells, they become rearranged during development. However, most genes in most organisms don't differ between different cell types, and therefore I won't be considering this possibility further. So that leaves us with 2 other possibilities. In the first case, we might be missing an activator, for example. But when the activator is present, the gene might be transcribed. Another possibility is that the gene might be remote from an anchor point in a loop. The anchor point might be a condensate rich in polymerases, and therefore the gene is off. But when the promoter is at the condensate, it might fire and the gene is transcribed. I'm going to argue that both of these are important. Now let's turn to something quite different, regulation, and ask what are the most important results in the field. I believe there are 2, and unfortunately the results are conflicting. Yamanaka took a mouse fibroblast and overexpressed just 4 transcription factors and converted it into a completely different cell type, a stem cell. So this, I would suggest, is indicating that gene regulation is relatively simple. GWAS gives the totally opposite result. Some background: it's an unbiased approach and therefore incredibly powerful. If we take any complex phenotype, which might be human height or the fibroblast or the stem cell phenotype, and look at the sequences in the population and ask which loci and how many determine that phenotype, the loci are known as QTLs, it turns out that there are thousands in the human genome. The expression QTLs are the ones that we're going to be mainly concerned with because those are the ones that are affecting messenger RNA levels. They rarely map to genes encoding transcription factors or even genes encoding proteins. So how on earth could Yamanaka's experiment work. They usually map to enhancers. Now, enhancers are mysterious motifs defined in many different ways. I'm going to use one definition which sees them just as active non-genic transcription units yielding enhancer RNAs rather than messenger RNAs. And I would point out that in man, there are many, many more enhancers than there are genes. So enhancers are just non-genic to use. EQTLs are just enhancers that target one gene, and similarly, one EQTL can target many genes that are often functionally related. And the targeting involves physical contact. Well, how do they work? This is one fashionable model. It openly lacks molecular detail. It explains functional relationships with difficulty, always at the post-transcriptional level, and no attempt is made to explain contacts. I'm going to argue that these two work together at the transcriptional level. Now let's turn to structure and ask, what are the most important players? Over the years, many have been discussed. CTCF is currently fashionable, but it's not in plants or bacteria, and therefore I will only consider it further in passing. These two fashions have disappeared completely. The everlasting structure are loops. Now, the first loops seen were all stabilized by the transcription machinery. And here are some early examples, with the contour lengths indicated, which ranged from the very short to the moderately long, and all sizes in between. Now, loops nowadays are usually detected by high-C, which is actually hopeless at detecting the loops that were detected earlier. Even micro-C misses most of the loops. Nevertheless, all of these current sequencing methods all confirm a central role of polymerases and transcription factors at anchor points, and so by implication as the stabilizing mechanisms in all organisms that have been looked at. And the highest resolution data that I'm aware of looks at the loci around these genes in mouse stem cells and concludes that about 70% of the genomic contacts involve promoters and enhancers. In other words, the transcription machinery. Contrast, for example, 4% involving CTCF. Here I list the known stabilizing mechanisms, and 3.5 of them involve the transcription machinery. Turn to another important area, complexity. I've already said that the fields are huge and fractured. I've already said that there are thousands of loci around the human genome that affect a phenotype of interest. So we need a wide guide through this complexity, and that will be provided by the theory of natural selection, which shows us that we can emerge from the primordial soup, as well as many other complex structures and functions, like birds, bacteria, and flowers. The simple can develop into the complex spontaneously, and this will be a recurrent theme throughout this presentation. Let's note in passing that anything found to be true of E. coli must also be true of elephants, and of course I'm going to have it both ways. In biology, there are always going to be exceptions. There are fish that can fly. So, to summarize the first section, I've argued that a gene is on or off due to differential factor binding and conformation. I've given you a big fact about regulation. It's both simple and complex. A big fact about conformation. The motifs that are the most important are going to be loops stabilized by the transcription machinery. And I've reminded us of a big guide, that of natural selection, which enables simple things to develop into complex things. In the next section, I'll go on to describe a classical mechanism that drives looping and clustering of the transcription machinery. Thank you for your attention.